Hello and welcome to the Future Medicine AI YouTube channel. My name is Harry and I'm the editor here at Future Medicine AI. Today I'm going to be sharing with you an interview with Jeff Lichtman, who is a Harvard neuroscientist working with Google Research to reconstruct the human brain connectome. This is essentially all of the neural connections in the human brain, and recently, in the last few weeks, they published some research reconstructing a single cubic millimeter of human brain, which doesn't sound like a lot, but it was in fact 1.4 petabytes of data to do this, and just an astronomical task. So Jeff delves into his work leading up to this achievement during this interview, and also gives us a peek into the collaboration with Google, and also his extremely exciting and ambitious future work to reconstruct volumes of brain tissue thousands of times larger, from mouse brains to even potentially in the future the human brain, which I ask him about. So I hope you enjoy, and I apologise that the conversation actually starts midway through our chat because the first bit was cut off due to recording issues, but I hope you enjoy, and I look forward to sharing some more exciting content with you very soon. So enjoy.
And I'm basically a one-trick pony. I've been doing that same experiment on ever larger and more complicated data sets, but doing it largely to understand how in development the wiring diagram gets established, because as you may or may not know, that early on the wiring diagram is quite different when mammals are born, including human beings, than the way it is later on. And a lot of the change in the wiring diagram is due to the elimination of a subset of connections and the selective enhancement of certain connections as other connections get pruned away. And so that's basically what I've been studying my whole life. And at the beginning, I was doing it with physiology, then with what we call XFP mice where a fluorescent protein was expressed in some subset of cells. And then I began crossing those cells to get multiple colors, and then finally made these mice called Brainbow mice.
The Brainbow mice.
Which are very, really amazing, but for very fine structure, they didn't have enough resolution. So then I switched over to serial section electron microscopy, which I had dabbled in earlier, and realized that this was a way to get enough resolution, and then partnered with Google just by accident almost. They were looking for a big project, and we had just finished a big volume, and they said they'd be interested in trying their hand at whether they could segment out machine learning, do all the stitching, alignment, segmentation, and ultimately synapse annotation on a big data set. And so we did the data set that you've just read about. And it's about 10 years of effort. So
it's absolutely astonishing. I mean, this is exactly the kind of thing that inspired me. I mean, I remember one of my first lectures, I saw the Brainbow, and I became mesmerized by that. And, it's these things like optogenetics and all of these really exciting things make neuroscience. I guess it's the sexier part of neuroscience, but it makes it's...
Yeah, I mean, it's interesting because I see such a fundamental difference between tools like optogenetics and tools like Brainbow. And maybe it's worth just saying a little bit about what I see as the fundamental difference. And that is that this is a tool that generates data but doesn't test hypotheses. It generates hypotheses, but it doesn't test them. Whereas optogenetics is basically to test an idea. You stimulate an area you think is important, or you block it, depending on which optogenetic probe you're using, and then you see what happens.
Absolutely.
Manipulation. Whereas Brainbow and the serial EM is really descriptive. We're not manipulating anything.
No.
I mean, if we are manipulating it, we're not doing it on purpose. We don't want to manipulate anything.
Yeah.
And then from this data, you look for things that are surprising. Things that are not the way it is in the textbooks. And that is how you move forward, is that those oddities you see force you to generate new hypotheses to change the way we think about the brain. Whereas optogenetics and tools like that basically say we know how it works. We have a model of how it works, and now we're going to do a test. And you only really publish those tests when they're compatible with your model. If they don't work, it's usually considered negative data. And there's an infinite number of reasons why an experiment might not work. So you only see the successful cases, but the successful cases only perpetuate the theories you already had. So it's not a very, for me personally, not a very effective way to move forward. So I'm very much in favor of descriptive neurobiology, just as Darwin or the Hubble telescope or the James Webb telescope.
I like
just
that.
looking at things, then you start seeing things, and then you have to come up with an explanation. And
Yeah.
usually it's based on what you've observed, not on what you hoped to observe.
Absolutely. And
It's a subtle distinction, but that's how I see.
Yeah, no, absolutely. I think that's a great way of putting it. And you did discover some quite interesting findings from you discovered some new neuron types, am I correct?
Yep,
and
neuron types and different strength, these super powerful, very rare connections, which we didn't expect. I think what we found was mainly proof of concept findings. That is, the main point of this study is to generate a data set that is so large that no human being or no laboratory can sort of say, we've picked all the high level results and now you can pick the bones if you're another lab.
Yeah.
The data set's so large, there's an unlimited amount of things you could do with it.
Yeah.
Of which we did a minuscule small number, and in several cases we've already found things that were not consistent with the worldview that's in the textbooks, and that to us suggests that there's probably a gazillion other things in this data set, and subsequent data sets that will be forthcoming, that will be transformative in the sense that they'll force us to have to reconsider what we think how the brain works. Yeah. So, for me personally, the most interesting result was the last result described in the text of the main results, which is that among the 99% of connections between axons and dendrites that are very weak, there is a very small percentage, somewhere between 1 in 500 to 1 in 1,000 axons make disproportionately large numbers of synapses on one of the many target cells that axon innervates. So an axon, and it's not just a special subset of axons, as far as we can tell, that any target cell we look at will receive a huge number of weak connections from axons, and yet there will always be one or two outliers.
Very interesting.
And if you trace those outliers, those axons don't always make strong connections. In fact, they also only make weak connections except on one or two target cells. So it's as if the nervous system has, in the cerebral cortex, two modes of action. One is related to things like temporal and spatial summation, which maybe you know about from your neuroscience background, where it's the synchronous activity of multiple axons that may be distributed in multiple places on a neuron that are necessary to bring the cell's membrane potential at the initial segment of the axon to threshold, so the cell can fire an action potential. That's the standard view of how information flows through the nervous system.
Yeah.
And that raises an interesting paradox that you don't see written about much, but it's pretty obvious that if only weak connections are there, and the way you get a cell to reach threshold is synchronously firing, let's say for the sake of argument, 10 axons that have to all release neurotransmitter at the same time. And that target cell, that cell, when it fires an action potential, only makes one synapse on each of its target cells. To get one of those target cells to fire, you would need 9 other axons that are synchronous with this one, each of which are driven by 10 axons themselves. So you need 100 synchronous axons to get one cell downstream to fire.
Yeah.
And if you're talking about a cell downstream another synapse, you would need now 10 times more of those, and each of them would require 10 synapses. So ultimately, if you had a relay that had 10 synapses in it, you would need 10 to the 10th axons synchronously firing to get that to happen. And that's more neurons than there are even in a human brain. So I don't think that is a very reasonable way to go.
Mm.
And so it is quite interesting that, in fact, there's another way. You could have a super powerful connection where one axon is enough to drive a cell to threshold. And then that cell, once it's driven to threshold, has one target cell where it has enough power to drive that cell to threshold.
Mm.
And that target cell has one output where it can drive its target cell to threshold. Then you have a fast path to get information, let's say, from your eyeball. If you're driving a car and you have to take your foot off the gas pedal and put it on the brake when you see a red light, you don't have to think about it.
Right.
There's no summation. There's nothing going on. You can be listening, talking to somebody, listening to the radio. It doesn't matter. It's post-cognitive. You're not even thinking anymore. And you have these reflexive behaviors that you've learned. Maybe that's what we're seeing in these brains is the things that haven't yet been learned, which is the substrate for all future learning.
Mm.
And then this, amazing subset of very fast, powerful connections, which are the memories that are now instantiated in the brain. So that to me is really interesting because I don't know of a better theory for how the brain can do things quickly once they're learned. And yeah, that would have maybe even some input and insight for the way one thinks about artificial networks.
Yeah,
and
absolutely.
They do something well, how you might do that, as opposed to just changing the weights of connections. You could trim away all but a small subset of them. So it's interesting. So
Absolutely.
that for me is an interesting result, but it was not the purpose.
No, of course.
It just came out.
Yeah. So tell me a bit about the collaboration with Google, because I find that very interesting, and how Google's technology must have, because I mean, the amount of data that you're talking about here, 1.4 petabytes of data, and just tell me how this collaboration with Google worked and how this has enabled you, how their technology enabled you to do this work, because...
I mean, it's been really a fantastically useful and wonderful collaboration in all respects. Google, I know, is a company and it makes money, but I'm dealing with a group there that is just very dedicated to solving hard problems, not for some commercial gain, but I think it's part of the DNA of that company that they believe their success is partly because they don't shy away from the hardest problems. This is how they learn to be a better company. And so for one reason or another, they were eager to do this project. I didn't offer them this project. They came to me.
Wow.
They knew we had big data, I guess, and they were saying that they thought they could handle this, but it was not trivial for them to do machine learning on 1.4 petabytes of data. Did require a lot of engineering on their end, and they did it. And yeah, then they did all the synapse annotation, hundreds of millions of synapses. And we're now doing many other projects with them because of this that are even more ambitious than this one. But
can you tell me anything?
They're great to work with. Yeah, I can tell you about them if you're interested.
Yeah.
One of them is we have done a connectome of a larval zebrafish. Zebrafish are larval zebrafish sounds like it's very immature, but this is a free-swimming animal that catches paramecia. Its visual system is good enough that it can chase around these small protozoa and eat them, and it can do many other behaviors. So although it's a immature fish, it's not immature in the sense that it can't fend for itself. It's a free-swimming, independent animal. And we did a connectome of an animal that had, at Genelia, the Howard Hughes site in Virginia, had already been studied by doing activity-dependent labeling with calcium imaging agents. All the neurons in the brain of this same animal, not the same species, but literally the same animal, had been studied as the animal went through 10 different behaviors.
Wow.
So there is this activity map of every cell's activity for all these behaviors. And then we took that same animal and we did the serial section reconstruction of the animal. And the goal is to superimpose now activity on top of the wiring diagram.
Wow.
So
That sounds
that project
spectacular.
is in process. The heavy lifting for us of imaging is complete, and now Google is doing the segmentation of the fish based on what we imaged, and that's largely complete, not quite complete, but it's largely complete. The synapse annotation is underway, and now there is the beginnings of trying to superimpose the activity map on top of the wiring diagram.
Wow.
And then even a more new project is that the federal government has funded here the first proof of principle projects to try to do a whole mouse brain's connectome.
What?
So instead of doing something that is a petabyte or two, to do something that is about 1,000 times bigger, an exabyte. So
What?
it's 1,000 petabytes.
And that's something do you think it's possible?
I sure hope so. We have $30 million to do the proof of principle with Google and a number of other groups. I'm the PI
Yeah.
of the principal investigator, but there's a lot of groups and a lot of different universities plus Google that are working on doing, for the proof of principle, only 10 cubic millimeters, something 10 times bigger than this data set,
Yeah.
but with techniques that we think can scale. We have to prove that
Yeah.
the techniques could scale. So it's just a matter of buying more machines
Yeah.
to get this to work on a whole. So we have 5 years to do the proof of principle. If we're successful, then presumably the government would go out and find the money, which would be a lot of money to do an exabyte connectome.
Wow.
That's the next big project.
And what's the main limitation of that? Is it the compute or is it the imaging?
Yes.
Yes.
Both.
So
I mean, everything about this, doing a petabyte, 1.4 petabytes was hard. I mean, everything about it was hard.
yeah.
But when you start doing a whole brain, then you even have to have the challenge of getting the whole brain well stained. That's not easy either. You have to have a well-stained brain, and then you have to not lose any of it when you start doing the imaging. And now you're imaging so much data, even things that happen very rarely. Let's say something that happens 1 every 10,000 times. That can be devastating if you're trying to do a whole brain and you lose a piece, so you can't connect the wires on both sides of that piece, because you're going to have to do about 80,000 sections. This was 5,000 sections. You have to do 80,000 sections of something much bigger. Yeah.
Because, I mean, before this, I wasn't aware of, being able to do image annotation on this scale with electron microscopy. I've been following, I don't know if you heard of Gubra. They're a company in Copenhagen, and they do some amazing light sheet microscopy, whole mouse brain imaging, and I find that mesmerizing. But this is just on a whole nother level.
Yeah, I mean, if this could be done with light sheet, it would be much easier. It's just that doesn't have enough resolution.
No, yeah, of course.
Usually the light sheet is at best a micron thick.
Yeah.
And here, we're dealing with things that are 100 times thinner.
Yeah. And looking to the future, what's your vision of it being done on a human brain?
A lot of people are curious about, well, if you're going to do a whole mouse, why not just do a whole human brain? And there's a whole bunch of reasons why that's impossible to do right now. One reason is the obvious one, that the digital data set would be closer to a zettabyte than an exabyte, so another 1,000-fold bigger.
Yeah.
So you would be talking about, instead of 1 million terabytes, 1 billion terabytes or something like that. It's basically equivalent to the digital content of the world in a year. So it's too much data. The other thing is how to get a really well-preserved human brain. the data that we're using here is from an operating table from a living patient.
Mm.
Once a patient dies, usually death is determined by brain death. When there's no more brain activity, the brain is already changing. And so you would like to get a fresh brain, but ethically, there's no way to get a fresh brain. And certainly a fresh, healthy human brain is harder to imagine. So I think for the time being, we are more than happy because it's a big enough challenge to do a mouse brain. And then I think maybe a more scientifically interesting question to ask is, at some point, we will presumably not be surprised by what we see in these brains. That is, we will have done enough in mouse brains that no longer are we shocked because everything we've seen before. That may be the best humans can do, by the way, when it comes to understanding.
Yeah.
Is not be surprised, even though we can't explain many things. And because we'll continue to do human samples, the same will occur. At some point, we're not going to be surprised. And then the question is, well, what's the upside of doing a whole human brain as opposed to doing little pieces if there's no longer any surprises?
Yeah.
And that's a really important question, because maybe to get the pathways that connect different parts of the brain, you don't need this kind of resolution. So you can get that in another way. And now you've looked at enough regions and you've looked at a whole mouse. So there's nothing surprising anymore. So maybe it would be just redundant. It's also, you're not going to learn anything new. It's also true that humans probably store in the wiring diagram a lot of their memories. And so everyone's brain is going to be different.
Yes.
So it's not that everything you're going to see in that brain is fundamental, because
I was thinking exactly that.
One-off. It's the unique experiences of the individual. So for all those reasons, I'm willing to put off until I come back in my next karmic reincarnation, because I don't think it'll happen in my lifetime to do a whole human brain.
No, but I think it was just it was just sort of the ultimate question. I can see from this, it's
it's not that I haven't thought about it, and I think other people have thought about it. And some of the people like Ken Hayworth at Genelia, who used to be a postdoc with me and now works on these newer techniques to scale up to do a whole mouse brain. I think he is interested in doing whole human brains, largely with the hope that one might be able to, upload someone's consciousness in silico, or even more way out, is broadcast it out at the speed of light into the universe and hope some advanced people somewhere.
I've not heard that one, actually.
Something.
Broadcasting. I've not heard that.
Reincarnate your brain.
Wow. Do you think that if you could do a digital twin, do you think that would replicate consciousness?
I don't. I think, part of the digital twin problem here is that a brain's activity is stimulated by its sense organs. And so you would really have to recreate everything, including the retinas and the hair cells in the ear. And they'd have to work, because the patterns of activity going through those wires is not implicit in the wiring diagram. It depends on the ion channels in each of these axons and the dendritic computations that are going on related to nonlinearities, related to the density of ion channels and dendrites, and how many neurotransmitter molecules are released at the various synapses. None of that is sort of easy to understand just by looking at a wiring diagram at one moment in time. So I think it's going to be very hard to, in the movies, you sort of upload the brain and then the computer screen says hello. But how does the brain actually type? I don't think it's never really clear. Those letters come out of the brain and onto the computer screen, because there is no interface there. So I'm a little skeptical that we're going to have that artificial intelligence is going to be replaced by human intelligence that's now in silico. I think that's unlikely, but maybe that I'm missing something, and I am old, so there's lots of new ideas there.
Have you had much interest in organoids? And
Yeah, I've worked on with Paula Arlata. We've worked on organoids here. She had an organoid paper that we participated in that where the organoid, if you shined light on it, the neurons would be activated because there were retinal cells in there connected to neurons.
wow,
And yeah, we did a serial reconstruction of the organoid to see if there were
oh wow,
synapses. And there were synapses in it, but it was so disorganized compared to a normal brain. It was very hard, you couldn't find anything recognizably that reminded you of a brain other than the fact
interesting.
that the synapses looked, but they were not, the density was very low and the cell shapes were very weird.
Yeah,
It was, after all, an organoid.
yeah.
So, but I think organoids, they're not going away. They're
Mm.
becoming very interesting, especially for looking at human cognitive diseases, making organoids from humans who have various forms of either adult psychiatric diseases like schizophrenia or developmental disorders like autism is a cottage industry that's being done a lot now,
Mm.
with the hope that something will be discovered that gives us some insight into what actually is physically wrong with these people.
Yeah, absolutely. And in terms of your I think it's fantastic that you've open sourced all of your data and all the information in, which I mean makes sense with you, like you said in the paper. There's so much that you it's a drop in the ocean what you've done. But what are you excited about for other people to do with your work, and or what are you hoping that people will do with the data?
Yeah, so I've already told you, my view of inductive science, that is hypothesis generating science, as opposed to hypothesis testing science. And I would like people to feel that there's nothing wrong with doing description. And so having data sets where you can describe, you can be an explorer. It's like this is a Galapagos island that is largely unexplored, and here is the boat ramp. Just get off the boat and just go in the woods. See what you can find. Maybe you'll find something fantastic.
Yeah.
And I think that's what I would like to see is people, including amateur scientists, not necessarily professional neuroscientists, just explore. For example, lots of amateurs who are not astronomers are fascinated by the James Webb.
I was just going to say it reminds me a lot of the James Webb Telescope. And
so imagine if there was a website somewhere where every picture they took of the universe is available and you just look around.
Yeah.
And here you can look around and click on something and render it in 3D, and you can click on its neighbor or an axon that's innervating that cell. It's amazing what you can do.
Yeah.
And the amazing thing for me is that here is 1.4 petabytes, and it feels like it's a minusculely small data set. Yeah, I'm sorry you were having slow time loading, but I think if you have a fast connection, even the slow loads, where hundreds and hundreds of cells are rendered, still load pretty fast. But if no cells are loaded, and you just go into the EM and click on a cell, it's only a second or so before that particular cell out of the thousands that are there will be rendered for you instantly, basically.
Is it possible
How they do that? I don't know.
Have you seen a render of the actual? Have they actually rendered the whole 3D thing in? The whole three-dimensional image in one view? Have you seen that?
We have made a render of it. One of the reasons their approach works is that when you want to render the whole volume in something the size of your screen, it's very low resolution. It looks great. And then if you zoom up, I want to only render half of it, it will use twice the resolution, but I'll show you half of it. And then if you want to zoom up even more, so the amount of data it's showing you, whether you look at the whole volume or just one dendritic spine, is the same amount of data.
Got you.
It's just a clever use.
Yeah.
So you always think you've got all this data there, but when you're looking at it so you can see the whole thing, it is 1.4 petabytes, but you're actually rendering it,
like a compressed version
of that data.
Yeah. Yeah, and this is very much off topic, but kind of well, it's not that off topic, but you mentioned your interest in space a couple of times, and I resonate with that. And I don't know if it's just a neuroscience thing, but are you quite inspired by the
I love descriptive studies of anything.
Mm.
I find paleontology, again, completely, there's no manipulation. All you're doing is digging up bones, reconstructing ancient animals. I find that very interesting. I think, you would call a person like me a naturalist, but it's basically the same feeling that people who are explorers have, which is my exploration is of the natural world. I'm more interested in that than looking at buildings, for example, or even being in an art museum. I'm very interested in the natural world, but whether it's bones or brains or, I don't know, a B for something out in the cosmos, but big galaxies.
Yeah.
They're all equally fascinating to me. It's just I happen to know a lot more about brains, so my questions are a little more in depth there. But I think fundamentally, I think there's a kind of person, and I'm just one of a gazillion people like this who are just curious about what the world actually is, and that, in my view, the best humans can do is describe the world until finally we've described everything. And we're far from describing everything. But that's very different from the modern scientific view that we don't understand something today, but if we work a little harder, we'll finally have the mechanism and we'll do the experiments and we'll understand it. I think understanding is overrated. I think it's almost impossible to understand a brain. It's just too complicated. It's
beyond comprehension.
The thoughts that humans have, even though our brain allows us to have thoughts, what comes out of our brains are far less complicated than the machine that's letting us think. So I think we're not going to get to a place where someday you say, I get it, I know why every synapse is where it is. I don't think that's going to happen. It's like explaining London or New York City. You say that you can't explain it. There's just, what do you mean understand London? There's so many things happening at the same time. And my view is, if you can't understand London, forget about trying to understand the brain, because the brain is far more complicated.
I think that's a great analogy. I think that's a brilliant analogy. Well, Jeff, thank you so much for taking the time to meet with me. I've really enjoyed the conversation, and
my pleasure.